And we welcome our session 4, Outlook for Drug Device Combination Products. Our speakers will be Dr. Christina Street, Senior Physician of the Office of Safety and Clinical Evaluation within Office of Generic Drugs. Then we have Dr. Jenny Wang. Oh, I don't have your picture here, that's why. Then we have Dr. Betsy Ballard, Medical Officer of the Office of Research and Standards, ORS and OGD. And then we have Dr. Jenny Wang, Visiting Associate within the Office of Research and Standards, ORS. And we will start with our first speaker, Dr. Christina Streets. Thank you.
Okay, good afternoon. So my name is Christina Streets. I'll be presenting with Betsy Ballard, the drug device combination products, methodologies for user interface evaluation. Today, our objective is 1, to provide key principles for comparative analyses and understand key comparative analyses definitions. 2, discuss FDA's experience with comparative analyses. 3, explain the human factors research being conducted by the FDA. And then we'll describe the taxonomy and how to implement it. So I'll begin. First, I'd like to start with some key definitions and information regarding generic drug device combination products. Therapeutic equivalence considers whether drug products are pharmaceutically equivalent, bioequivalent, and whether the products are expected to have the same clinical effect and safety profile for the conditions of use specified in the labeling. Therapeutic equivalence is also expected for generic drug device combination products and takes into consideration whether the end user would be able to appropriately administer the drug product without the assistance of a healthcare professional or without receiving additional training prior to use. And that's when the generic product is substituted for the RLD, the reference listed drug. This does not mean that the two products need to be identical, but the differences must be acceptable and allowable under the abbreviated new drug pathway, the ANDA pathway. Excuse me. So how do applicants and how does the FDA determine whether these differences are acceptable? The draft guidance, comparative analyses, and related comparative use human factor studies for drug device combination products, sorry about that, submitted in an ANDA, which was published back in January of 2017, provides applicants with guidance on how to conduct comparative analyses to assess differences in their proposed generic device compared to the RLD. The guidance describes 3 comparative analyses to be conducted. The labeling comparison should be a side-by-side comparison of the proposed labeling compared to the RLD, and for our purposes, focuses on the delivery device constituent part of the labeling, and that would be in the prescribing information, in the instructions for use, and then any other related labeling. In the comparative task analysis, the applicant should take apart and analyze all the differences, all the potential differences in the use process for the proposed generic product when it's compared to the RLD. And then the physical comparison should evaluate the physical design differences of the proposed product to the RLD, and it should include both a visual and tactile examination. To conduct a comparative analysis, it's important to first understand some of the key definitions. When we talk about the user interface, we're considering all of the components with which the end user interacts. This could include the delivery device, this includes the delivery device constituent part, as well as any controls, displays, product labeling, and packaging. The critical task is defined as a user task that when performed incorrectly or not performed at all, would or could cause harm to the patient or user, where harm includes compromised medical care. Identification of critical tasks takes into consideration the indications for the drug product, the users, such as patients, caregivers, or healthcare professionals, and the environment and other conditions that might influence the importance of a particular task. External critical design attributes are those features that directly affect how users perform a critical task that's necessary to administer the drug product. So what have we seen in terms of submitted comparative analyses? Between January 2018 and December of 2023, we received over 1,000 comparative analyses for ANDAs. The majority of the submitted comparative analyses based on the route of administration were injection products. That's followed by ophthalmic products and then oral products. We also see nasal, inhalation, and ODUC products, as well as products we've classified here as topical, vaginal, and rectal products combined. What are the possible outcomes when conducting comparative analyses? The draft guidance mentioned earlier provides detailed information regarding the 3 possible outcomes. 1, no design difference. 2, minor design difference. And 3, other design difference. No design difference is exactly what it means. There's no difference between the proposed product and the RLD. Minor difference means that there is a difference in the user interface of the proposed product compared to the RLD, but this difference is not expected to affect an external critical design attribute, which I remind you is a feature that directly affects how a user performs a critical task. And finally, other design differences. These are differences in the user interface design that may impact an external critical design attribute and therefore may impact a critical task. And the important part there is that it may impact. There we go. So what does it mean when a product's determined to have an other design difference? How does it affect approvability as an ANDA? If it's determined that the product has an other design difference, it still may be approved as an ANDA, but it may require additional evaluation. We need to consider how the design difference could impact any errors, question whether it changes the clinical effect or safety profile of the proposed product compared to the RLD. And we take all of this in consideration of the RLD as well as a specific ANDA. We look at the context of use of the product, such as the end user population, the environment of use, the urgency use, and also the frequency of use. Sorry about that. So how often do we see other design differences that are unacceptable? Of the over 1,000 comparative analyses I mentioned previously, there were 83 applications that had other design differences that were unacceptable. This is approximately 10% of comparative analyses. This slide does not show other design differences that we deemed acceptable, though. So these are ones in which the other design difference was unacceptable. The other thing that we'd like to make note here is that often throughout the review cycle, we are able to make recommendations so that these other differences are, made to be minor differences or no difference potentially. And so we only send a complete response letter if we're unable to rectify the issue during that cycle. The majority of products that had other unacceptable differences were injection products, accounting for over half of the applications. Oral products made up a quarter of the applications, and then ophthalmic products and those products under the topical, vaginal, rectal were the remaining applications. When we identify these unacceptable design differences and issue a deficiency, we recommend that the applicant consider modifying the design of the user interface to minimize any differences with the RLD. If the applicant does not intend to change their design, then we recommend that the applicant submit additional data to support or justify that the other design difference will not alter the risk profile of the proposed product when it's substituted for the RLD. Examples could include in vitro studies or a comparative use human factor study. The comparative use human factor study has been an area of research interest for the FDA, and I'll pass on the presentation to Dr. Ballard to discuss further. Dr. Ballard?
So just to get an idea, a sense of the audience, a show of hands for those in the audience, and I think you can vote online. How many of you have submitted and/or conducted a comparative use human factor study? So yes, you have, no, you haven't, or you have no idea what I'm talking about. So how many have submitted? 1, 2, 3. So how many have not? The vast majority, I'm assuming, have not. Okay. That's kind of what we suspected, because this seems to be a pain point when you have to consider doing a comparative use human factor study because you have another design difference. This has been considered a pain point, and so it's one of the reasons why the research program was started. So this is just a quick timeline because I wanted to show sort of the relative the recency of this whole area of research that's the FDA is conducting. The draft, as Dr. Street said, went out in 2017. In 2020, at the start of COVID, OGD stood up what's considered the device evaluation team. And we work in the pre-ANDA space to advise industry and companies on design differences for their proposed DDCP and help them work through some of the issues and how to obtain data that will allow them to then go on for approval. So as part of that, in 2021, the requests went out for proposals to study the comparative use human factors arena. And that, while some of you in here are probably familiar with the traditional human factor studies like the validation studies or usability studies, the comparative study is different because it requires you to compare your product to a reference product rather than comparing it amongst its using your device design and having users evaluate it and make iterations and design differences based on that. So it's a little bit different. 2 grants were awarded that year. One was entitled User Interface Designs for Generic and Reference Listed Drugs, and that went to Battelle Centers, the Public Health Research and Evaluation. And the other grant that went out was for the development of a taxonomy to help identify these things, and that was granted to the University of Detroit. So the first grant, the one that went to Battelle for the user interface designs for generic and versus RLD combination products, had 2 aims. It wanted to develop enhanced methods for threshold analysis and categorization of user interface differences. And the second aim was to try and establish effective methods to assess these other design differences. Unfortunately, that grant terminated early and there are really no outcomes to report. The second grant, the development of the taxonomy from the University of Detroit, had 3 aims. The first one was to develop a body of knowledge of key stakeholder perspectives and existing strategies that people were using to assess the user interface design differences. The second aim was then to use that information and build on it and develop a visual taxonomy to assess the user interface for combination products. And finally, to use that taxonomy and actually apply it in a comparative process using a reference drug and a generic drug. So what is a taxonomy? When I took this project on, I had no idea what they were talking about. But it's a method for organizing specific concepts and creating a common vocabulary for those concepts, so that when we talk about something, everybody understands what we're saying. And the key thing that the University of Detroit collaborators wanted to do was link that design feature to a specific task and risks that are associated with that task. Okay. So what did they actually accomplish? They did accomplish aim 1. The interviews were completed with the key stakeholders. They interviewed human factors engineers, CEOs, and a variety of people, and they performed a literature search to see what was available in the public arena on comparative use human factor studies rather than the traditional human factor studies, and prepared a publication which is currently undergoing peer review. For aim 2, they did develop a taxonomy, and that's what I'm going to talk about in the subsequent slides. And they validated this taxonomy in a small inter-rater reliability study. They are developing a case report using their taxonomy and fabricated data to show how this taxonomy can be used to determine whether your design differences are other or minor. Aim 3, unfortunately, which was applying this in a broader concept, was not completed. So what is the taxonomy? So it's when started with the combination product categories, and they used a 21 CRF 3.2E, and they used that to help define what a combination product was. And then after talking with the agency, they were able to prioritize which products they would look at in more depth. They then you looked at each of those devices and developed task analysis for each device that was very detailed. And they also looked at developing use error analysis for each of those tasks and for each of those devices. And they linked that to the URAA. They then linked that risk and the task back to a specific design feature and came up with a taxonomy. And what they started with, guys, it's not advanced. Okay. What they started with was basically look like this. And, you know, it kind of reminds you of the elemental chart in chemistry. They basically identified two major areas. One was labeling and one was user interface. They then went down and broke each of those down into certain specific categories, and then subcategories and descriptors. So the further down you get on the table, the more specific you're looking at. So on the and this is sort of a breakdown, and it probably shows a little bit better one of those particular categories and how you can trace it down. When you're looking at your device, you would start at the descriptor and work your way back on up through the thing. But they categorize each design attribute by color, size, shape, texture, and material. So very much like our guidance says, you want to look at the shape, size, and orientation of each product. And they looked at the literature and found what were the risks associated with each of those particular design features. They realized that shape and form, so a minor difference, they decided would be a change within a subcategory, whereas another difference would be a change in a critical design feature, so that it would be, the difference would be higher up or in different categories altogether. So this was obviously a very cumbersome way of doing it, and they developed a spreadsheet that incorporated each of these tasks and walks the user through a series of questions that delve further into the hierarchy and allow you to classify it. So it looks something like this. And you can see this is for a blood pressure cuff. And so you can see that they have it broken down by the group. The tasks are all there, and each one is assigned a group. And it's color coded, so it's very easy to look at, and you can just get a very quick visual as to where your differences are and where you might have to concentrate on looking at things further. They ultimately wanted to use the results of this hierarchy, this spreadsheet, and you would do one for your RLD and one for your test product, and you could quickly identify where the differences were. And then the goal of the project was then to take those differences and assign each thing a risk and come up with like sort of a relative risk value and produce what they are calling a determination report. And so in order to be a minor design difference, that it must link to a non-critical task. It cannot add any new or increased potential for harm. It cannot add or subtract or eliminate a task. It can't change the action required to complete that task. And it must fall within the same descriptor within the taxonomy. If a design feature does not fit those, then there's potential that it would be considered an other design difference. And they propose that because this could be, because each of the ultimate value of this would be the risk that would be associated with this, you could use this to help you justify why your design differences may or not be acceptable for an ANDA submission. But at this point in time, the acceptance of this report or any report such as this in an ANDA submission is a bit premature because none of this has been finalized or validated. Where do they see where do our collaborators see the future going? Well, clearly, even a spreadsheet is not the easiest to work with, and so a web-based taxonomy tool would be ideal. And they want to further take the step one step further and use those results to help you identify minor versus other. And that'll be a risk-based task-oriented assignation. They need to apply the taxonomy in a larger study. using a variety of participants, so not just human factors engineers or engineering students, but also industry reps and potentially even regulators to see in an interrelated reliability how often do we agree on the classifications. And that will allow them to continue revising and updating the taxonomy tool. So that project terminated in June of this year. And so what ongoing research does the FDA currently have in the human factor space? And what we have is an IDIQ, or an indefinite delivery, indefinite quantity. And the goal, because of all the feedback we've been getting from the agencies about trying to do these comparative use human factor studies and all the problems that are associated with them, they decided that the task one for this IDIQ would be to actually conduct a human factor study the way that our guidance suggests that we do, and following the recommendations that we give to industry, so that we could identify where exactly the problems are, what are the issues, and how can you move past them and resolve those issues, so that information can be published and be available to industry to help you do a better job conducting a human factors potential study. And the other interesting thing about this would be, once we have this data, then we could take it and look at evaluating with potentially different statistical methodologies. And that's very exciting at this point. So in summary, the taxonomy, it can be a powerful tool because it provides a common language. It can assess, the way this one was designed, it can assess the level of risk associated with the design differences, but it needs to be implemented in a larger study of the comparative process. The Office of Generic Drugs continues to fund human factors research. The IDIQ went out in March of last year. The devices are being selected, and the comparative use human factors study protocol is being developed. But what I really want to draw your attention to is the broad agency announcement that will go out in November of this year for FY 2025 to encourage anybody who might be interested in coming into this comparative use space to go to that webcast, which is, I think, November 7th, and certainly submit your proposals for consideration. And with that, we'll go on to the challenge question. Which of the following is true regarding other design differences? A comparative use human factor study is always required. The design difference may impact a critical task. They preclude approval under an abbreviated new drug application, or the applicant must modify the design difference to minimize it. And so anybody want to, do we have the results from the poll? They're not coming up, so that's what I'm asking. Okay, the answer is the design differences may impact a critical task. And challenge question number 2, which of the following are current research projects that the FDA is conducting? The taxonomy development, the user interface design for generic versus combination products, a comparative use human factor study, or there's no current research in this space? And the answer for that is we are in the process of conducting a human factor study. And with that, I'm going to turn it over to Dr. Wang, who's going to present her topic.
Hi, good afternoon, everyone. My name is Jenny Wang. Today, the topic I'm going to talk about is approaches to analyzing comparative use human factor studies. So this is a disclaimer. So the learning objectives for my presentation today, the first one is to provide the overview of the current use of comparative use human factor studies to support other design differences. And the second objective is to discuss the analysis approach for the non-inferiority test in comparative use human factor studies. First, I will talk about the current use of human factor studies to support other design differences. So in the FDA's draft guidance, the comparative analysis and the related comparative use human factor studies for a drug device combination product submitted in an ANDA, we can see, like, in this procedure at the very beginning, during the early stage of the development, applicants are encouraged to minimize the differences from the user interface for the reference for the RLD product. And then applicants can conduct the threshold analysis, including labeling comparison, comparative task analysis, and the physical comparison of the delivery device contingent part. So the result from the threshold analysis may be no design differences or differences in design. For the differences in design, if that falls into the categories that's other design differences, applicants can either modify the user interface to minimize the differences or to provide data to support the differences, just as my colleague just presented. Applicants can provide, can conduct a comparative use human factor studies to support the differences. So for the human factor studies, it's designed to confirm that the use error rate for the critical task for the proposed generic combination product is not worse than the corresponding use error rate for the RLD. So here is a procedure of comparing the error rate of test product and the error rate of the RLD product through the non-inferiority test in human factor studies. There are mainly like 4 steps. The first step is to determine the allowable margin D by which the error rate of the test product could exceed the error rate of RLD product. The second step is to estimate the study sample size, considering assumed error rates and the margin D. The third step is to observe error rate for the critical task during the human factor experiments. And the fourth step is to perform the NI hypothesis test. So I will introduce the details of each step in the following slides. So for the first step is to determine the allowable margin D by which the error rate of the test product could exceed the error rate of RLD product. So the value of this margin D will differ between products. It's depending on the indications and the clinical consequences associated with failing to perform the critical tasks appropriately. And the acceptable margin D should be decided in consultation with FDA before the study is conducted. The second step is to calculate the study sample size, considering assumed error rates and the margin D. So in the FDA draft guidance, it provides an example using the Tango method to calculate some power simulations given selected sample size and the alpha level equals to 0.05 and the allowable margin D equals to 12.1, just as this table shows here on the right side. So the Tango method used in this example is the widely used method to calculate confidence intervals for the differences of 2 proportions in a paired design of clinical trials. To utilize this Tango method to calculate the confidence intervals, usually there are several parts of information required, like the number of subjects who completed the R tasks successfully but had errors in the T tasks, and the number of subjects who completed T tasks successfully but had errors in R tasks. Also, you will need to have the total number of subjects and the confidence level. We want to note here is that the Tango method is just one of the options for the analysis of the human factor studies. So the third step in this procedure is to observe error rates for the critical tasks during the experiment. To conduct the experiment, first of all, we will need to define the critical tasks. And then we will need to observe the error or success results of subjects for each critical task. Like on the right side, this table here, this is an example data set for a given critical task. We can see like each row can represent the result for each subject. So the first column represents the results for the RLD product, and the second column represents the results for the test product. We may use like this kind of binary results, like with the 1 means like subjects complete this task successfully, and the 0 means the subject may have encountered some errors or failed this task. So after the experiment, the fourth step is to perform the non-inferiority test. To conduct this non-inferiority test, we will need to, based on the experimental data, we will need to calculate the confidence intervals for the difference of error rates between the test product and the RLD product. Then we need to compare the upper bound of this confidence interval to the allowable margin D. So if given the alpha level equals to 0.05 and the upper bound of the 95% confidence interval is less than the margin D, then the null hypothesis is rejected and the non-inferiority is demonstrated. So for the data analysis part for the comparative use human factor studies, in addition to these current recommendations in the draft guidance, FDA continues to conduct internal research to facilitate drug development and mitigate regulatory burdens for the human factor studies. For example, like our group has been working on examining like the potential alternative data analysis methods and approaches for the human factor studies. We are working on evaluating the performance of different alternative methods. So the applicants are also encouraged to propose alternative data analysis methods and the study designs for the human factor studies. So here, there are just like some examples of the alternative data analysis methods to consider. For example, non-parametric methods may be considered, like the bootstrap-based methods, or Bayesian methods can also be considered to estimate the distribution of the target population's performance with the test and the RLD products. Applicants are also advised to discuss proposed alternative data analysis methods and study designs with FDA before initiating the comparative human factor studies. There are programs available for the discussions, like the Model Integrated Evidence Industry Meeting Pilot. So this is a new pilot program to offer meeting opportunities to applicants who intend to use model integrated evidence or novel data analytic approaches for BE establishment in their ANDAs. Also, the pre-ANDA program for complex generic products, also appropriate program for the discussions. The ANDA applicants for complex generic drug products can request product development pre-ANDA meetings to help clarify regulatory expectations early in their product development stage. Here, I would like to acknowledge the help and the support from my colleagues and the leadership from the Office of Research and Standards of OGD. Thank you. So here is our first challenge question. So in the FDA draft guidance, comparative analysis and related comparative use human factor studies for drug device combination products submitted in ANDA, the comparative use human factor study is recommended for every application of drug device combination products. So this statement is True or false? Yeah, it's false. That's good news. So the second challenge question is, in the FDA draft guidance, so which of the following analysis methods is recommended for the non-inferiority test in comparative use of human factor studies? We have some options. First is Tango method. Second is bootstrap method. Third is Bayesian method. And the option D is the guidance doesn't recommend the analysis method for an eye test. Yeah, the correct answer is D. So Tango method was just used like in an example in the guidance. It's only one of the options for the analysis. Applicants encouraged to propose like novel analysis methods for human factor studies. And the FDA are open for discussions. Yeah, now we will go to the Q&A session. I would like to welcome Christina and our other panelists to join us on the stage. Thank you.
Thank you very much, our panelists. And we will welcome our two. Did somebody fall? No? We welcome our two additional panelists, Dr. Sumesh Chattopadhyay, lead mathematical statistician with the Office of Translational Sciences. And we also welcome Dr. William Chong, who's the director of the Office of Safety and Clinical Evaluation within OGD. Okay, everyone. I'm so happy to see everybody still here at the end of the day. Welcome. Online folks, I hope you're hanging out with us as well. Any in-house questions? Acknowledged. Let's go to our online questions, please.
First question for Dr. Betsy Ballard. The taxonomy seems like a complicated way to address a complicated analysis. Does the agency think this is an improvement from a review standpoint compared to what you are seeing in submissions?
I think the purpose of trying to develop something like this is very often the determination by a review team or even an individual reviewer can be very subjective. So it might vary from team to team or from reviewer to reviewer. And if we have a more qualitative type of method to apply, the hope is that the industry will see less of that variation and have a more consistent response in our recommendations.
Thank you. Next question, please.
Right. Next question addressed to either Dr. Ballard or Dr. Streets. A risk-based assessment of human factors is very dependent on an assessor's understanding of risk. How does FDA educate its assessors regarding appropriate assessment of risk? How does this assessment differ based on the end user and context of use for the proposed product?
Yeah.
Thank you for the question. So in terms of assessment of the risk, we really look at it from a clinical standpoint. And when we look at the context of use, we're looking at who is using this product and what is the intended use for this product. So a product that is used emergently is going to have a different level of risk than one that's used chronically. And in terms of how we handle that as a division, we really try to communicate and to the best of our ability come to an understanding of what is considered a critical task and what is a potential other difference if we're identifying other difference. Our division looks at the comparative analyses, and we really try to discuss with each other. So for anything that may have where there may be some confusion about what the risk is.
Thank you. Next question, please.
We have a question for Dr. Wang. Is there a recommended allowable margin D in the FDA draft guidance?
There is not a recommended allowable margin in the guidance. So the 0.1 allowable margin was used in the example. But like for each product, I think the allowable margin is a case-by-case situation. It's related to the indications and the risk for like each critical task. So like this allowable margin should be discussed with through the consultation with FDA, and the applicants recommended to provide the justifications and the evidence for their proposed allowable margin. Yeah, thank you.
Thank you. Online question, please.
Another question for Dr. Wang. What kinds of internal research is FDA conducting to mitigate regulatory burdens for CUHF studies?
Thank you for the question. So as I mentioned a little bit like during the presentation, our group have been working on evaluating the performance of different potential alternative data analysis methods for the human factor studies. We are evaluating like how well each method can control the type 1 error and type 2 errors through simulations. Yeah, thank you.
Can I add something?
Yes, please.
So, I mean, I think, Dr. Wong's group is doing a lot of work looking at some of the just different statistical approaches. And, we heard her talk that there's lots of different ways people can approach the studies. A point that I think I wanted to emphasize from Dr. Street's talk was, we see lots of comparative analyses. We get lots of device products submitted. I think over 1,000 in the, 6 years since we implemented the draft guidance. Less than 10% of those get to the point where the design difference is something that's unacceptable. So I think that's first something to note that, really we're talking about a small set of products compared to what we're getting in OGD. And the other thing I think it's worth mentioning is, we spend a lot of time today talking about the work that we're doing to support the development of the design of comparative use human factor studies, the statistical approaches and things. But I do want to emphasize, in the guidance, one thing that I think often gets missed is that, we're open to other approaches to justifying those differences. So if a comparative use human factor study sounds very burdensome, and that's something we've certainly heard from applicants, and as Dr. Ballard mentioned, that's part of why we're funding the research that we're funding. That doesn't necessarily mean that is the only way to validate and justify your other design difference. So I think that we're open to both considering alternative statistical methods, understanding the rationale for your justification of your non-inferiority margin, or if you think there's some other way you can justify why a design difference is acceptable under an ANDA and can be approvable. I think we're open to hearing those justifications and thinking about them. And I know Dr. Streets has mentioned, when we find those things in our assessment, sometimes we will ask questions, ask for more information, more justification, and potentially changes, different things. And we do want to work with you to get these products to the market so the American public has access to them.
Thank you. Next question, please.
Next question, again for either Dr. Ballard or Dr. Streets. How does a generic drug sponsor submit a comparative threshold analysis for an approved product where a PFS is proposed, but the approved RLD is a vial, or that there are no approved generics with a PFS?
Okay, so this is something that we run into frequently. And I think when you're looking at that, you just have to consider what the design features are and what their purpose is. So while it may seem impossible to do a physical comparison of a prefilled syringe versus a vial, it's really important to just provide the information that compares them, and then the labeling as well, and then also, the task analysis. It's not impossible to do. You just have to submit the information and the comparison. So I can't think off the top of my head of a side-by-side, but if there's not an applicable side-by-side component to the comparison, then you can state that.
Yeah, I would just add that when we see this happen, and we've had it happen a couple of times where the RLD is a vial and they want to come in a prefilled syringe or they want to come in an auto injector. And I think in that situation, you really do need to come in early and come in on the pre-ANDA side so that you can start to get our feedback early as you continue your device development, because there may be issues that we're aware of that we can help in guiding you through that process. But I think the main thing is going to be when you have that is the task analysis, because are you eliminating tasks? Are you making it easier for the end user? So you need to keep the end user in mind and the environment of use, because if the task analysis shows that there may be some advantages, we may be able to do that. There's certainly many products that were originally approved as a vial with a syringe, with or without a prefilled syringe as part of the kit, that have now come in as prefilled syringes or as auto injectors even. So it can be done, but we strongly recommend that you come to us early in the pre-ANDA space so that we can iron out those differences.
Okay, next question, please.
Another question for either of you, Dr. Streets or Dr. Ballard. Is it possible to address differences that are considered other through label changes and IFU and not only with device UI changes?
So you cannot label your way out of a difference. But again, we're not saying that every design difference needs to be changed. So again, there's additional information that you can provide to justify that the design difference is not likely to affect the clinical safety or, the clinical effect or the safety profile of the product when it's substituted for the RLD. So the change, you do not have to make the change, but you can't just change your labeling in order to make that okay.
And there's certainly data that can be obtained and extracted from other areas. Certainly what's out in the public arena in terms of real world data for known use errors and things like that, that can be brought in to show that, to maybe justify your design difference. You can conduct your own study that may show that there's no difference, even if it's not a formal CUHF. So it's not necessarily that you have to do a CUHF if there's an other design difference and you don't really want to modify your design, or let's say you can't modify your design because of patent issues with the RLD. There are other data sources that you can use and bring in to help justify your design differences. And again, this is where it becomes very important if you're looking at something like this to come in both in the pre-ANDA space so that we can advise you early on, so that when you do get to the point of your ANDA submission, some of this has already been resolved and you know which way you have to go.
Okay, next online question, please.
This question came in addressed specifically for Dr. Ballard. If the RLD is not available in the proposed device, how does a generic drug applicant submit justification through a comparative threshold analysis?
Okay. What we usually recommend in this situation is you have to compare to the RLD, and that can be done. Certainly the labeling, you can usually get a copy of the RLD labeling, even if the product is discontinued, and you can use that to do your side-by-side labeling. You can get instructions for use and use that to do your side-by-side labeling. Where the issue becomes stickier is when you're looking at the actual device itself, because the RLD may no longer be available, but usually there will be a reference standard that can be used not only for the drug product, but also for the device issue. And you can go ahead, and we have seen submissions where they have used the physical comparison to a reference standard as additional supplemental information to support the use of their generic, because the RLD is no longer available. But we still recommend that you try and make a best effort to get the RLD product, and if not, at least use the RLD product for the labeling and the task analysis.
Yeah, I would emphasize that part, that you should be starting with the RLD as much as you can find, as much information as you can find. Photos of the RLD labeling, those sorts of things. and that some of these supportive or supplemental information from a reference standard or another generic version referencing the RLD can be part of that sort of other information that helps us evaluate and understand, is your proposed product going to be usable in the marketplace? Are people going to be able to use your drug product in place of the RLD, which, recognizing in this situation, the RLD is discontinued?
Okay, thank you. Next online question, please.
I have a question for Dr. Chong. How do you think the ongoing research work that OGD is supporting will help the development of generic combination products?
Thank you for that question. I think we've already heard a little bit about how some of this can be helpful. I think Dr. Ballard's initial response to the question around the taxonomy is one that potentially can be helpful just to make sure we're all speaking the same language, acknowledging the initial questioner's framing of it is complex, but it is a starting point, and that might help get us to a place where it becomes more second nature to do these kinds of assessments, and we're using the right terminology so that everyone is understanding when we're questioning a specific design difference, we know what we're talking about. So I think that's one place. I do think that as Dr. Ballard mentioned in her presentation, designing a comparative use human factors study, I think will be very educational and informative for us to understand where are the pitfalls, where are the challenges, what are the things that we can advise on to make the assessment of these other differences easier going forward in the future. And I can't speak to the statistics. I'm not that bright, but I think alternative methodologies for analysis of data, I think, is something we can certainly look at.
Yeah, and also I'd like to add that as Dr. Ballard mentioned in her speech, that there are grants, so we are working on those things also. And for the methodology, actually, I want to tell you that FDA doesn't recommend any specific method for the analysis. So anything that is appropriate for the design is acceptable. Acceptable in the sense that if it works for that particular design, then we can accept that. So actually, the word or phrase alternative method actually is a misnomer in that sense, because there's an alternative to what? We don't have a prescribed method, basically. So anything that is acceptable, that is appropriate, may be acceptable. So I just wanted to stress on that. And you can, the key is to come to FDA early, so that if you have any method that is not widely used, if you want to use that, or if you have any innovative ideas, you can come and we can discuss that.
Yeah, thanks for the correction on the terminology there.
Okay, thank you. Next question, please.
One more question for Dr. Wang. Could you give an example of how non-parametric methods may be used for CUHF studies?
Thank you for the question. So for the non-parametric, I would like to give example like using the bootstrap-based methods. So bootstrap resampling may be used to through the resampling, it can be used to estimate some attributes of the target population for the statistical inferences. Then based on this method, maybe the confidence interval of the non-inferiority test can be calculated. Yeah, this is a brief example. Thank you. Yeah, so much if you want to add more.
Yeah, but whatever method, again, there may be other non-parametric methods, and but if you think of something new, just come to FDA and we can discuss that. Because Jenny just mentioned one particular one.
Next question. No more questions. All right. And let's anybody in the house has any question? They're excited about Axon? No? Okay, let's thank our panel. And okay, we do have our closing remarks, so please remain seated. And you, the audience, please remain seated. And those online, don't sign off yet. Okay, so for our closing remarks, we're going to welcome Dr. Larry Lee, who is the Deputy Director of Operations in the Office of Pharmaceutical Quality. Dr. Lee is currently the Deputy Super Office Director for Operations and oversees research, quality surveillance, policy, quality assurance, and administrative operation functions in the Center for Drug Evaluation and Research's Office of Pharmaceutical Quality. Dr. Lee has been with the U.S. Food and Drug Administration since 2005, serving as a regulatory scientist, team lead, associate director for science, deputy office director, and deputy super office director of science. He has provided exemplary leadership in developing OPQ science, research, and testing programs to support quality assessment, inspection, surveillance, and policy. So help me welcome Dr. Lee.
So I promise I will try to finish my concluding remark very quickly so you guys can get out of here, all right? But first of all, really thanks for the wonderful introduction. I do want to say that like generic, I think I was actually in Office of Generic Drug before. And I want to really emphasize that our generic is really built upon science. And the research is really a critical piece of our generic program. And our research continues to support the advancement of a generic drug product development from both a product quality perspective as well as bioequivalence perspective, because you really need both elements in order to ensure the safe and efficacy of the generic drug product. And as you probably already hear throughout today, our research supports the quality assessment of various complex generic applications, also not just quality assessment, but also the bioequivalence assessment in terms of formulation approaches, manufacturing, performance, and demonstration of BE. And I want to share with you my perspective actually developing a generic, especially for the complicated one, is not an easy task, right? It's not like just like, okay, you put this and give it to patient and then look at the clinical endpoint, you get it done. No, I think it's more complicated than that. That's why our research always focus on the critical aspect as well as the innovative aspect of the drug development. This includes the analytical method for for assessing pharmaceutical equivalence and bioequivalence of complex generic drug products. Just give you an example, like developing assessment criteria for formulation and microstructure sameness of the complex generics, such as semi-solid topical products, as a demonstrate. Really, it's not an easy task because, right, like you are trying to use those circuit to replace the clinical or the PK endpoint study, which really can help to speed up the development of generic drug in this area. And as discussed today, the agency continued to support the development of more predictive in vitro testing to establish the bioequivalence as well as the quality standard for these complex generic drug products. As I want to point out that our office actually worked very closely with the OPQ, the Office of Pharmaceutical Quality, work really closely with the Office of Generic Drugs to really continue to overcome a lot of our scientific challenges in this area. And the result of the research is not just to, publish the PSG. Actually, the outcome of the research also help us to advance the policy as well as in the future regulation for the generic drug products. And then most likely, I think the industry may not realize that. And this research outcome also help to speed up and facilitate more streamlined assessment in both of our bioequivalence area as well as product quality areas. So we remain committed to maintaining transparency with regard to the expectation and communication in the research area, how we actually translate those scientific outcomes to the regulatory use between the agency and the industry. And our standards for improving the quality of the submission and enabling an efficient development and review of generic drugs remain consistent. And we pretty much remain committed to really work with our industry to facilitate and accelerate the generic drug development. And thank you for all the excellent presenters as well as my colleagues for your continued contributions in advancing generic drug development. Remember, generic drugs actually about 90% of the market. So I think it's really thanks to all your hard work and really try to think outside the box, continue to make the high quality as well as equivalent generic drug available to the US patient. And thank you for your attention.